And thank you all for coming. So before I start the actual seminar, I just want to talk very briefly about MPG Ranch, because probably most of you in this room have never heard of us. So we are located in the Bitterroot Valley on a private ranch, a pretty big property. We conduct research related to restoration ecology, invasion biology, plant-herbivore interactions. We also do a lot of tracking of bird migrations, primarily raptors, and do a lot of bandings of songbirds as well. A big component of what we do is educational outreach, and we have several thousand people come to the ranch every year to visit. And if you're curious about what we do and want to learn more, we have a website as well. So this ranch was purchased in 2009, and one of the first things we did was to go out and do a pretty careful survey of the ranch using a grid point system. And what became apparent pretty quickly was that this ranch was inundated by invasive plants. And here I'm showing the three primary invaders that we have: cheatgrass, Bromus tectorum, spurge, leafy spurge, Euphorbia azula, and spotted knapweed, Centaurea sturbii. And this is the distribution of these three species on the ranch. And unfortunately, cheatgrass is the most abundant species on the ranch. So I'm relatively new to invasion biology, and one thing that has blown me away is how these evolutionary naive species can come into and outperform and outcompete locally adapted plants. And when they do that, they often increase productivity. And this is data showing Morgan McLeod's master's work that was published. And what she did was to go out and just measure productivity in invaded and adjacent native grasslands. So she targeted independent invasions, actually several sites of cheatgrass, spotted knapweed, leafy spurge, and she also added a fourth invader that isn't as problematic in our area, Potentilla recta, which is sulfur cinquefoil, but we certainly have it, and it's certainly one to keep an eye on. And for the three major invaders, she saw a significant increase in productivity associated with these invaders. And in some cases, especially with leafy spurge, it was more than a doubling of productivity on average. So one hypothesis is that these invaders go in and they invade disturbed ecosystems that also have higher nutrient availabilities. But we've shown in an experiment that we set up, a common garden experiment, where we actually planted these four invaders and remnant native community, or not remnant native, mixed native communities, where we've measured productivity. And we see the same pattern. These are the mixed native communities with representative forbs and grasses, and sulfur cinquefoil, cheatgrass, knapweed, and leafy spurge, mirroring really what Morgan found in the grasslands that she monitored. And this also suggests that this is not only then invaders invading already disturbed grasslands that have higher nutrient availability. They're also, under common garden conditions, able to produce more than native plants do.
Can I stop there?
Yeah.
Can you go back to that? I'm not sure what the graph means. What is it?
Oh, sorry. Yeah, well, thank you for pointing that out. This shows productivity in grams per square meter. So we actually go out to these communities and cut the above ground biomass. And then, so basically in the native communities that we see an average of 300 grams per square meter. And when we go out to the leafy spurge communities in this common garden, we see more than a doubling of that.
What you mean by productivity is like the...
Everything that is above ground. So we go out basically and put a frame that is 0.5 by 0.5. So we measure a quarter of a square meter, and we cut everything above ground when they've reached maximum productivity, which is usually end of June.
So you're saying, like, compared to native plants, you have twice as much leafy spurge? Is that what you're trying to say?
Yeah, the biomass in leafy
The
spurge
volume.
communities.
Oh,
Not,
okay.
yeah, the biomass, just the weight of leafy spurge is more than twice as much.
Okay.
Is that clear? Thank you for
I'm
asking.
kind of naive about
No,
this,
no,
so
it's all
I-
good. Whenever anything is unclear, I'd rather we talk about it now than later. So that's all good. So, you know, I was hired to try to understand what is the potential role of the soil ecosystem in this. When we have these invaders, it's pretty obvious what happens above ground, but we know relatively little about what happens below ground in terms of both microbial communities and associated ecosystem processes, in terms of nutrient cycling and soil respiration and whatnot. And there are the reasons to believe that soil biota is involved in the success of invaders. There are at least 4 hypotheses in invasion biology that suggest the role of soil biota. The first one, the most prominent one, is the enemy release hypothesis. And you've probably heard about it, but what that predicts is that exotic plants are successful because they escape their native enemies, such as herbivores and soil pathogens, for example, that occur in their native range. The biotic resistance hypothesis suggests that in some native grasslands, there could be some resistance towards invasion, and that could be driven by the fact that there are some native plants that are closely related to the exotic that is coming in and therefore harbor similar soil pathogens. Then there are 2 hypotheses that are related to mutualisms, and I will cover those in more detail later. So basically what I want to try to cover today, and hopefully at least partially provide answers to, are these 4 questions. So do microbial community composition differ between invasive and native plants? Are differences invader specific and predictable? Meaning if we go to multiple invasions, do we see similar shifts, and are they invader specific? Are they associated with shifts in ecosystem processes? And maybe most relevant for you guys is, what are the implications of this for restoration? So I'm going to start by talking about arbuscular mycorrhizal fungi, and to limit the time I'm saying that, I'm going to abbreviate it as AMF here. I don't know if you've heard about these before, but they're fascinating and really abundant soil fungi. They form this separate phylum within fungi, all of fungi, called Glomeromycota. They are extremely common and colonize about 90% of all plant families, about 80% estimated of all plant species. So it's a lot more common for a plant to be mycorrhizal than to not have this association with these fungi. And that has led to these statements that plants don't have roots, they have mycorrhizes. And it is a symbiosis. It's not always beneficial, but the basic exchange is that the fungus gets carbon from the plant. It's an obligate symbiont, so it gets all the carbon that it needs from colonizing these plant roots. And in return, it delivers nutrients and potentially other services to the plant. And because it's in such high abundance and occur in this root-soil interface, it can also have large effects on ecosystem processes. This is what it looks like. It's a microscopic association, so we can't just pull up a root and see that it's mycorrhizal. This is taken from a petri dish and under a dissecting scope, so we see roots here. And they are in auger, so that allows us to see these hyphae growing out from the roots, and we see spores forming here. If we take a section of that root and stain it using a blue stain, obviously roots are not blue, but we use a blue stain which works because the fungus absorb more of that blue stain than the background roots. So these fungal structures that occur inside the root are actually then prominent, and we can look under the microscope and quantify that root colonization. So these are typical structures that these fungi form. These are just vesicles where the fungus store the carbon that it gets from the plant. And then we have exchange sites here where it gets gives phosphorus to the plant in return. And sometimes these roots can just be full of the fungi. So going back to these 2 hypotheses in invasion biology that relates to mutualism, there's one that we call the enhanced mutualism hypothesis, and that was coined by Kurt Reinhardt and Ray Calloway at UM. And it's based on the idea that some invaders have a higher dependency and benefit more from this association than native plants. And there are some examples in the literature. For example, spotted knapweed has been shown to become more competitive when it's inoculated with these fungi. There's another example of Canada goldenrod, which is native here, but it's actually exotic and invasive in Europe and parts of Asia, one that Robert has worked a lot on, which shows that when it invades, it changes AMF community composition in ways that make it more competitive. The other hypothesis is quite the opposite, and it's called the degraded mutualism hypothesis. And that is from Vogelsang et al. In 2005. And what they're saying is that invasive plants are predominantly non-mycorrhizal, have a low mycorrhizal dependency. And when they invade, they're also then going to reduce the abundance of AMF, which is going to hurt native plants that are more dependent on these fungi. So you can see they're quite opposites of each other, these two hypotheses. There's some support for that. Garlic mustard, we don't have it here, but in the Midwest and Northeast, I believe they're pretty bad, and that is a non-mycorrhizal plant. So there's some really elegant studies showing reduced colonization by native tree seedlings here and lower productivity of those.
So mutualism basically is talking about the symbiotic relationship. That's what mutualism refers to.
The symbiotic relationship just referred to a close relationship, symbiosis, living together, and that could be both parasitic, but it could also be mutualistic. So the effect of that symbiosis, yeah. So in this case, we assume that in most cases, it's going to be beneficial to both partners. California grasslands, a lot of invaders in California grasslands are non-mycorrhizal or have a low colonization, and they've shown that invasion by those reduce AMF abundance. And looking at it from a biogeographical perspective, comparing plants when they're in their native range compared to the invasive range, there seemed to be a selection for decreased AMF dependence in at least a few species. Okay. So the first thing I wanted to do when I came and started with MPG in 2009 was to simply go out and ask, do we see changes in AMF communities associated with these invaders, and are they invader specific? And are they predictable? Meaning if we go to independent invasions of leafy spurge, will we see similar shifts in the AMF community? And I did that in collaboration with Søren Rosendahl at Copenhagen University and Sean Gibbons and Philip Ramsey at MPG Ranch. So we targeted 6 different locations in and around Missoula, and at each location we had these 4 communities: cheatgrass invasions, knapweed invasions, leafy spurge invasions, and then remnant native communities. So that meant that we had replication of each plant community. And in each community then, we sampled roots, and from the invaded communities, we only sampled the target invader. So in cheatgrass communities, we only sampled and characterized cheatgrass roots, whereas in the remnant native communities, we targeted representative grasses and forbs that were present. And then we characterized the AMF communities using molecular techniques, and I'll outline how we do that. So we usually start, because these are microscopic, we can't just look at the roots and determine what species are there, because they are microscopic, and we can look at root colonization, but that only tells us abundance. It doesn't tell us the species identity. So really the only way to do that is to extract and amplify DNA and sequence it. So that's what we do. We start by either roots or soil, in this case soil. We extract the DNA and amplify selected DNA, either bacteria or fungi, or specifically arbusculomycorrhizal fungi, using specific primers that only amplify that group. Then we send it off for sequencing. Sequencing techniques have just, exploded in the last, 5 years. So we get like millions of sequences back, which has actually forced us to have to learn bioinformatics and computer programming, which I'm not personally good at, but I have people that I work with that are. And then finally, we match these sequences to curated databases to figure out who's actually there. And from these different communities in and around Missoula, this is what we find. I'll walk you through this ordination. This is taking all that multivariate data set, the whole AMF community, and putting it in two-dimensional space. What that means is that, you know, these everything that has the same symbol here is an AMF community from a Spurge community. So all the black squares here represent AMF communities from particular Spurge invasions. So the closer two symbols are, the more similar the AMF community is. And then we can overlay other variables that we measure in the soil, for example. So what we see here is that Spurge communities, Spurge invasions, harbor more similar AMF communities than a Spurge invasion that may only be 50 yards away from a cheatgrass invasion. So there is some structuring force here going on that depends on the plant invasion. And then we can see also when we measure soil nutrient availability that nitrogen, for example, nitrogen availability in the soil points straight at Spurge invasions, meaning that nitrogen availability is also higher in Spurge invasions. One interesting thing here is that if we look at this, the AMF communities associated with Spurge and cheatgrass are furthest apart, meaning that they are the most dissimilar from each other. And it's interesting because they are also the two plants that are most dissimilar from each other, in that Spurge is a perennial, deep-rooted forb, and cheatgrass is a shallow-rooted annual grass. So from this, we can say there is something going on with plant invasion and AMF communities, that when we have specific AMF communities that occur in Spurge invasion. From this, we don't know if Spurge actually caused these changes, or if Spurge may be invading areas where a particular subset of the AMF communities also thrive. What we also found, which was a big surprise to me, was that when we're looking at AMF richness, the fungal richness and diversity, I had hypothesized that that was going to be highest in the remnant native communities because they were also the ones that harbored most plant species. And we think that there is this relationship between plant richness and AMF richness. That is not at all what we found. In fact, we found that the highest richness of AMF were in the two were associated with the two forb invaders, knapweed and Spurge.
So richness is occurrence, diversity is the number of species?
So richness is just counting the number of species that we have. Diversity takes abundance into account. So it's both richness and abundance. So it's a combined measure of the two. I really like that you ask questions when we go along. It's a really good way of doing it. So keep doing it. So in this case, you know, I've had a lot of people asking me, do you think that is a co-invasion, that leafy spurge somehow bring specific AMF with them when they come to these areas? I don't think, at least in our case, that that is what explains the higher richness, because we see site-specific and rare taxa. So it's almost like the native communities have lost some of the richness that was there. And I'll come back later to why that might be.
Excuse me.
Yeah.
What is richness?
Richness is, in this case, you know, like it's based on sequence similarity. So we have all the sequences, millions of sequences, and we bin them. So if there are 97% or more similar to each other, we call it an operational taxonomic unit, because we don't really know what a species is and where the distinction is.
So it's not particularly rich.
You know, AMF is quite amazing that worldwide we might only have 700 or 800 of these OTUs. We have 300 of them in culture. And that's quite amazing, right? Given that they can colonize 80% of all plant species. So the richness of AMF is not particularly high. So to have 24 in a leafy spurge invasion, it's not necessarily that low.
And diversity again is?
It's a combined measure, so you take into account the abundance. So because obviously you have a few of these species that are really abundant and some of them that are really rare. So it's going to be more driven, the diversity is going to be more driven by the abundant species. Yeah. So looking at abundance, again, scoring these roots that we collected and looking at percent root colonization, just focusing on this first graph, again, with the native communities, cheatgrass, knapweed, and spurge. We see, again, like we had a higher richness and diversity, we also have a higher root colonization associated with these forbs, and cheatgrass is the lowest. So not only do cheatgrass and leafy spurge have the most similar AMF communities, they're also very similar in the abundance of AMF that they can maintain. And then we have a different way of looking at AMF abundance in soil, and we see a similar pattern. It's not the same, but it's a similar pattern. So I would say with this, at least in our system, that we have some examples of the degraded mutualism hypotheses, and we have some examples potentially with the enhanced mutualism hypotheses, at least in terms of the abundance that they generate. We don't know necessarily from work we've done that leafy spurge and knapweed specifically benefit from AMF, but other people have shown that in other research. So, of course, when you do something like that, you start to wonder, like, is what we're finding just an anomaly, or are there broader patterns that support what we're finding? So we decided to go to the literature and do a meta-analysis, which is basically just where you extract data from published research, and then you analyze that data to try to synthesize. So we had 3 primary questions with that. We wanted to know if root colonization differed, broadly between native and invasive plants. Is there a directional change with when plants become invasive? Do they respond differently to AMF? And then do AMF influence competitive interactions between native and invasive plants? And I did this together with, again, Philip Ramsey at MPG and Rebecca Bunn at Western Washington University. She's the one who's an expert on running these analyses for the meta-analysis. Okay, so just briefly, what is a meta-analysis? Again, we extract data from published research, and because it's not very informative to compare absolute values because it depends on the environmental conditions where the experiments were conducted, you have to relativize the data. So what we decided to do here, we calculated a ratio between the invasive and native. So taking root colonization of the invasive over the native. So doing that, you can see, like, if it's 1, the ratio is 1, there's no difference in root colonization or responses between the native and invasive plant. If it's less than 1, meaning that the native one has a higher root colonization than the invasive, and opposite, if it's above 1, that means that the invasive plant has a higher root colonization or respond more to AMF than the native plant. So what did we find? Well, overall, we found we found something, but there was no directional change when we compared like invasive plant and native plant as a whole. So there was they didn't differ in root colonization or responses, and AMF did not have a directional effect on competitive outcomes. However, when we started looking at functional groups, remember what we found in our first study was that the forbs seem to generate higher abundance and have a large effect on the AMF community, whereas cheatgrass, the grass, seem to reduce it. And we see the same thing when we look across all published work today. I'll walk you through this figure. This is, again, like no difference here. And we see when the native is a forb and we compare it with an invasive forb, there's no significant difference. However, when the native is a grass and the invasive is a forb, the root colonization is higher with the invader. Contrary, if the native is a forb and the invasive is a grass, it goes the other way. So the native has a higher root colonization than the invader, whereas if they're both from the same functional group, there's no difference. So there really seemed to be a functional group signal here rather than provenance, like meaning that if it doesn't really matter if it's an invasive or native, it just matters if you're a forb or a grass. Okay, in terms of responses though, we see somewhat of a provenance signal. So here we have invasive forbs, invasive grasses, and again, like here 1, meaning that here we compare inoculated plants that were colonized compared to non-mycorrhizal plants. If it's higher than 1, that means that mycorrhizal fungi actually benefited these plants. We see that the only one that really benefits significantly are the native forbs. And the native grasses are trending downwards, whereas the invasive plants don't seem really to care all that much. The other thing we see here is when we're looking at competitive interactions, mycorrhizal fungi really matters for the native plants, more so than the invasive forb. Here we didn't have any invasive grasses in all these studies in competition to include that as a factor. So you can see here when native forbs are grown in competition, they really benefit from AMF, whereas the grasses are actually hurt. Probably because if they're competing against the forb really benefits, and that means that they become a stronger competitive, and the grass is then hurting from that. The other thing that this indicates is that the responses of native plants, like basically going from negative to strongly positive, is much greater than the exotic plants. So it seemed to matter overall more for the native plants than the exotic plants. And we don't really know why that is at this point. So it's something we're currently exploring, but it is a pattern. So to summarize this first section, when we have support for both these hypotheses, both of them are probably occurring, but it can be explained and predicted based on the mycorrhizal status and the functional group identity of the plant. So far I've talked about AMF. What about bacteria? We're also interested in bacteria because they obviously perform a lot of important functions in a soil ecosystem. They're involved in most nutrient transformations that occur. So looking at, oh, that is a terrible, terrible pixelated picture. Okay, I apologize for that. This is together with Jack Gilbert with the Earth Microbiome Project. I don't know if you've heard about it, but it's a really cool project where they characterize microbial communities all across the world. And again, Sean Gibbons. We went out and collected soil from a lot of different plant communities. You can see the green is natives, reds are cheatgrass, blue are knapweed, and yellow is leafy spurge. And basically what I want you to take away from this, again, it's a survey, it's not from the experimental gardens, is that there seem to be, even for bacteria, this plant functional group response. And I say plant functional group response because these days, a lot of the native remnant communities are not high in forb abundance. And why is that? I think part of the explanation is that we've used a lot of forb-specific herbicides. So we go out and we have sprayed with Tordon, and that is beneficial for example, ranchers. They want to get rid of the forbs because the cows eat grasses. So what we're left with is grass-dominated remnant grasslands. And I think, and that is backed up by a lot of the surveys that we've done, plant surveys. So it's really like a predominantly grass response and forb response, even for the bacteria. Interestingly enough, we go back to the experimental gardens here. After 5 years, we also see that these plant communities separate the bacterial communities. So they structure the communities in different ways. And it's important to have these experimental common gardens because then we can start talking about causation. Just going out to the field, we can survey the communities, but we don't know if the invader actually caused those changes, right?
Did you feed the plants in your garden?
Feed them? No. No. It was pretty high because this is former ag land, so it was pretty high in nutrient availability, unfortunately. But again, like you can see, this is just the means of the microbial communities from the 5 replicate, in this case, leafy spurge communities. So they all, you can imagine like 5 different symbols here clustering down here. And again, like the most dissimilar bacterial community is cheatgrass. So again, the same thing as we're seeing with AMF communities, we see with bacterial communities. These invaders really have a large effect on both fungal communities and bacterial communities, and they are predictable. So one thing we did with the bacterial data is based on some idea by Noah Fearer. where we can look at bacterial phyla and kind of infer function. So he divides bacteria into oligotrophs and copiatrophs. And the oligotrophs are bacteria that can use more recalcitrant sources of carbon. They tend to grow slower. They turn over slower. Whereas the copiatrophs are, fast growing. They utilize pretty simple sugars and whatnot. And I like to use this analogy. You can think of them as kind of like the donut versus bran muffin. That's a mental good way. Oh, I forgot to say, and what we see with this when we do that is that not only do the composition change or differ a lot between leafy spurge and cheatgrass, they also differ on the phylum level, so that the bran muffin bacteria are higher in abundance with cheatgrass communities, whereas the donut bacteria, they're higher in abundance with leafy spurge. And that is important because it has implications for nutrient turnover and leakiness of systems. So we wanted to explore that and say, okay, we base that on community composition, but are there actually functional differences in these primarily bacterial communities, but in this case we're looking at the whole decomposer community from these experimental plots. So what we did was to go out to these experimental plots, collect soil, bring them back into the lab, and add a common resource. And in this case, it was just leachate. Basically, you make a tea from native litter. So basically what these decomposer communities will see in the fall. And then we had these 5 different soils. It was 5 reps of each, because we have 5 reps of each. You can kind of see it here, the cheatgrass communities, 1, 2, 3, 4, 5. And then we added this common resource, and then we asked, do we see differences in how they respond to this? And the response we measured was just soil respiration. So we measured CO2 emissions from these soils under incubations in the lab. It's a highly controlled environment, but all of these soils are exposed to the same resource, and we just wanted to see if there was a signal that they respond differently. What we predicted based on the community composition was that, again, the cheatgrass and leafy spurge communities would be most dissimilar from each other, that maybe the cheatgrass having these like bran muffin responses would have a slow initial response and maybe increase in how they utilize those resources, whereas leafy spurge would maybe just crash, use the easily accessible carbon in this substrate and then just crash. And that is hard to see, huh? That is indeed what we found. A lot of those things have also come away. This is time, so we measured respiration over time, and it's compared to a water control. So basically here, this is cumulative respiration over time, and you can see this exactly what we predicted, that cheatgrass communities just slowly take a slow, it takes them a while to get going, but then the respiration increases over time, whereas this is really hard to see. But leafy spurge, has this initial bump. They respond to it, but then they can't really utilize those resources. And then the respiration is negative here because it's lower than what we're measuring if you just add water. Oh, that's what happened. Oh, there we go. So we were wondering what might drive this. Of course, this is just taking soil that has been cultured by these plants for at this point 5 years, when we did this experiment. So, with mycorrhizal fungi, I think it's just based on host quality, and it's carbon allocation that these plants allocate to the fungi. But what is it with bacteria and potentially other decomposers such as fungi in the soil? What do they respond to? They probably don't respond to carbon allocation from a living plant, but they're saprotrophs, so litter is probably important here. So when we're actually looking at litter, we're taking these litters at a dead plant matter at the end of the season, and we're analyzing that. We see large differences specifically in carbon-nitrogen ratios. So the higher the carbon to nitrogen ratio, the more recalcitrant the litter is. And you can see here the two most dissimilar plants are cheatgrass and leafy spurge. And so you can imagine if you for 5 years have been cultured by Leafy Spurge, that is a pretty readily available litter that turn over easily because it has a low CN ratio, that will structure the bacterial communities to be able to utilize those resources. Why is this important? Again, I think it's important because it changes the function, it changes nutrient turnovers in these communities and leakiness. A lot of these bacteria that are more donut bacteria, they're not very good. They're just like boom and bust cycles. So that means that a lot of these nutrients that become available can then leach from the system. So now I want to talk briefly about what we're seeing in terms of nutrient availabilities in these systems. This is again revisiting Morgan's data for her thesis. Again, in these paired communities, she went out and also measured nitrate availability in these communities. And what you see here, except for Potentilla recta, it is a substantial increase in nitrate availability, even in cheatgrass communities. So in this case, I am not sure because she did field surveys. So in this case, I was not there when she selected those communities. It's possible that cheatgrass just follows Leafy Spurge, for example, and nitrate is already available and cheatgrass didn't create those conditions. We don't know. We will only know from the experimental plots. But that brings up the question, where does this nitrogen come from? And we don't know. I mean, one possibility is that, as I said initially, that these invaders invade disturbed areas. We know that disturbance facilitate invasion, and they may also then have higher nutrient availability, so the invader may not do that. It's also possible that they associate with free-living or endophytic nitrogen fixers. None of these are legumes, obviously, so they don't form rhizobia. But we're learning more and more about these endophytic nitrogen fixers that can live in bark of trees, and it has been shown in an invasive grass in the south that they associate with nitrogen fixers. Or a third possibility is what we call priming, where the invaders allocate more carbon below ground, which means that the microbes will switch from being carbon limited to nitrogen limited. And that means that they're going to go out into the soil and start accessing recalcitrant nitrogen that is bound in soil organic matter. And there is an enormous amount of nitrogen bound in soil organic matter that for whatever reason the native plants don't utilize. We don't know the answer to that. It might depend on the invader, but that's where we're going with our research now to try to figure that out. This is what we're finding in terms of nitrogen availability in the experimental plots. So this is, again, going out into the soil in June where all plants are actively growing and measuring nitrate availability. And this is over 2 years, so the blue bars is 2016 and the purple bars are 2017. And as you can see, there's year-to-year variation, and the error bars are large. So there's variation between plots for some reason. But overall, especially with the two forbs, we see higher nitrate availability. Whereas cheatgrass, it seemed to depend on the year. But for the most part, they tend to be higher than the natives. So they do change nitrogen cycles. We don't really know how yet. So to return to the outline, I feel like we've shown that microbial communities differ between native and invasive plants. They seem to be invader specific, maybe even like functional group specific, or at least we can make rough predictions based on functional group identity of the invader. And that these changes are also associated with shifts in ecosystem processes. So the last thing I want to talk about today are the consequences for restoration and what we're learning about that. One thing with restoration is that we use herbicides. We use lots of herbicides. And this returns to earlier what I said about forb-specific herbicides and how we've used a lot of that on our landscape, which has changed a lot of our native communities and reduced forb abundance. We were interested in picloram, which is really tordon. It's the active ingredient in tordon. And that is a forb-specific herbicide that we've used a lot on the ranch. We were interested in the below-ground effects of this herbicide. We wanted to know if those effects, if they are there, if they are directly mediated or directly, the herbicide is directly affecting the soil microbial communities, or if it's mediated by a shift in plant community composition. So in order to do that, we went out to 3 different areas on the ranch. In each community, we targeted knapweed invasions and either spray them with Tordon, 3 reps per community, or just spray them with water. And we did the same thing with the grass-dominated native communities. And the reason we added both communities is that we didn't expect any change in the plant community when we sprayed grass-dominated communities because it's a forb-specific herbicide. And that would then allow us to look at the direct effects of these herbicides, if any, on the soil microbial community. So what did we find? Well, first of all, spraying with Tordon or Picloram changed the plant community. Not surprising. It's meant to kill knapweed, and sure enough, that's what we see. So here we have the native communities, knapweed communities, and over 2 years, 2012 and 2014. And what we're seeing here is that there's hardly no spotted knapweed in the native communities. That's good, because we selected them to not have any knapweed in them. But in the community that was sprayed with water, we see pretty high abundance, almost 30% cover of knapweed, and that is reduced significantly, almost eliminated when we spray with Tordon. The other interesting thing is that when we spray with Tordon, it's actually replaced by bulbous bluegrass, which is another exotic grass that is similar in many ways to cheatgrass. So you get rid of one invader and another one comes in. That's what we think of as invasion meltdown often. On a side note, 3 years after we sprayed, couldn't really see that we had sprayed in the first place. So that just emphasized that you can't just go out and spray these communities. You have to do something else as well. The residual time of Tordon is about 2 years, so that's why 3 years. There was a lot of seeds in there, and the residual effect had worn off, and knapweed just grew back. So, but what we found was that because knapweed was replaced by bulbous bluegrass, that was a really drastic shift in AMF host quality. So just looking at the root colonization of knapweed is almost 80%. So 8 out of every 10 root segments that we're looking at are colonized by AMF. If you go down to the native grasses, it goes down to 20. And then bulbous bluegrass is even worse. It's like cheatgrass that way. It harbors very low abundance of these AMF. So basically what we're finding 1 year after these applications is that, again, I'll walk you through this, there is no effect on the native communities, meaning that Tordon doesn't have a direct effect on AMF communities or abundance. But, you know, it is an indirect effect here. So this is the communities that were sprayed with Picloram when we had that shift from knapweed to bulbous bluegrass. And we see that the AMF abundance is reduced more than half of what it was within a year. So there are rapid decreases of AMF when we spray with this forb-specific herbicide. And it's directly an effect of shifts in host quality.
How come there was such a decline in the knapweed with the control?
I don't think there is. This is a knapweed of control. It has a high. It's not significantly higher.
On that left,
yeah, here is the sprayed one.
on that left figure from 2012 to 2014.
Oh, here? That's bulbous bluegrass, yeah. So knapweed is about the same in the control, and you can see in 2014 is starting to come back. So this is 2 years, so the residual is just starting to wear off. If we had done this survey in 2015, there would have been no difference. I couldn't visually see differences when I went out. So a big thing with restoration, and you've probably come across it, everybody says we need to inoculate with AMF, you know, and there are a lot of products out there. A lot of them are bad, you know, because they don't have viable spores in them. And I hope I've convinced you that before you spend a lot of money to inoculate with AMF, you really need to ask yourself, who was the previous invader? If it was Leafy, Spurge, and knapweed, Well, you probably recovered a lot of AMF that might have been lost otherwise, and richness and abundance might actually be higher than degraded adjacent native communities. Whereas if it's cheatgrass or a non-mycorrhizal host, maybe you would have a benefit from inoculation. But there are ways to figure out what the inoculant potential is in a soil. It's a quite easy way. If you're interested in doing it, you can email me. If you have a greenhouse or just a good light in your house, you can do it. You just have to send the root samples to me to look at them. But there are ways that you can look at that. So the last thing, promise, that I'm going to talk about is like, what are the consequences for native plants that are growing after these invaders? You can imagine, like, if you're planting a native plant after cheatgrass, maybe root colonization is pretty low, whereas if you're planting it after a knapweed and leafy spurge is pretty high. If the root colonization is informative of shifts in abundance, which we think that it is, and we have some data to support that. So one thing we did a few years ago was to plant blanket flower. I'm sure you're all familiar with this beautiful native forb. It's highly colonized, and it's actually quite dependent on these fungi. We planted them in all these plots, and then we looked at root colonization. We just harvested them and scored them for root colonization about 6 weeks after we planted them. And we get this. So this is the same plant species, so it's not based on different plants, but it's based on different plants that are growing next to this blanket flower. So again, what we're seeing is that it's highest in leafy spurge and lowest in cheatgrass. So it really matters for the root colonization and potential growth and success of these native plants who the previous invader was. And we're seeing that these patterns roughly are correlate to what we saw in terms of internal colonization of these invaders and soil colonization of these invaders. Same thing, we planted a lot of different plants in these different communities. We tried to protect them from mice and grasshoppers. That's why they're in cages. I don't know why mice would like to eat, cheatgrass seedlings, but they do. So we need to protect them somehow. And then, so we planted all the invaders and 2 natives, again, blanket flower and blue bunch wheatgrass in all these communities. And then we just measured growth. How well do they do? And I'm just showing you the effect of the native plants. And this was, again, after growing for 2 months. So this is blanket flower biomass. Again, the taller the bar, the bigger the blanket flower is, or blue bunch. When they're planted into native communities, cinquefoil, oh, sorry, these are wrong. Oh, no, they're not. So cheatgrass communities, knapweed, and then leafy spurge. So what stands out here? Well, first of all, like blanket flower and blue bunch really like leafy spurge soil. And I think that is due to the higher nitrate availability that we have with spurge. Spurge is an amazing plant, and I'm actually quite fascinated by it, because if you look at the shoot nitrogen concentration, it's similar to a nitrogen fixer. This plant, I don't know where the nitrogen is coming from, but it harbors a lot of nitrogen. The nitrogen availability in the soil is higher. It has higher productivity. We have no idea where that nitrogen is coming from. And as you can see, it's biologically relevant. When you have a blue bunch wheatgrass seedling growing in leafy spurge, it's 800 milligrams tall. When it's grown in a native, it's less than 50. Oh, yeah.
So could this extra nitrogen come from prior land use?
Not in this case, because this is all in experimental plots.
Okay.
So they've been growing there for 6 years, and the conditions were the same before. Yeah. Yeah.
So you said if spurge is colonized more by mycorrhizae, right?
Mm-hmm.
That helps fix nitrogen, right?
No. Mycorrhizal fungi helps with phosphorus acquisition,
Okay.
but does nothing really for nitrogen. Yeah. So I think the nitrogen, when we measure nitrogen availability in leafy spurge soils, it tends to be higher. But I think the nitrate availability is not really showing the available nitrogen of these plants, because we see so much greater responses when we're actually growing plants than the chemical analysis would indicate. But look at this, so we have these 2 native plants, right? And then cheatgrass. And look at the y-axis here. It's almost 10-fold difference, right? So yeah, it matters for native plants, but they'll all be outcompeted by cheatgrass, because cheatgrass loves nitrogen. So that is another, indication with restoration. We plant native plants, and then you have this invasion meltdown, that there is another invader, often cheatgrass. We're actually seeing a reduction, at least in the Missoula area, of knapweed right now, and we think that it's due to the biocontrol actually doing something. What is replacing it? Cheatgrass. It's not native plants coming in, it's cheatgrass. Okay, last slide. So I hope I've been able to convince you that both bacterial and fungal communities change in predictable ways and invader-specific ways with invasions. I hope I've also been able to show that a lot of ecosystem processes change with invasion, and they are also invader-specific. So we can't simply say that invasive plants do this and native plants do this. No, it depends on the invader, and we have to start looking at the traits of the invaders, because then and functional groups, because then it will all start making sense, and we can start making predictions. If we just try to lump every invader into one group and expect them to behave the same, I think we're going to fail. And then finally, that these changes that we're seeing have potential effects on restoration. And I'm hoping that what we're doing and learning about the changes that we see with these invaders can help inform restoration practices. And the ultimate goal with these experimental plots is that we're probably going to have them going for a few more years, but then we're going to try to restore them to see if it matters. Maybe when you plant the native plants, they come in and they change the bacterial communities back, and it doesn't really matter. But we can also measure what time scale that occurs in. This is just an urge to everybody to think about the below grounds. It's so obvious what happens above ground, but I think to really fully understand invasion and to increase the success of restoration, which is pretty dismal, in my experience, we need to take the whole ecosystem into account and understand it. So we've had questions along the way, and this ran pretty long, but I think we have maybe a few minutes for questions, if you have any.
First of all, thanks so much.
Oh, yeah.